Recombinant DNA and gene cloning. Recombinant DNA molecules are DNA molecules formed by laboratory methods of genetic recombination such as molecular cloning to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in biological organisms. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure. They differ only in the nucleotide sequence within that identical overall structure. The DNA sequences used in the construction of recombinant DNA molecules can originate from any species. For example, plant DNA may be joined to a bacterial DNA, or human DNA may be joined with fungal DNA. In addition, DNA sequences that do not occur anywhere in nature may be created by the chemical synthesis of DNA and incorporated into recombinant molecules. Using recombinant DNA technology and synthetic DNA, literally any DNA sequence may be created and introduced into any of the very wide range of living organisms. Proteins that can result from the expression of recombinant DNA within living cells are termed recombinant proteins. Expression of foreign proteins requires the use of specialized expression vectors and often necessitates significant restructure by foreign coding sequence. Molecular cloning is the laboratory process used to create recombinant DNA. It is one of the two widely used methods along with polymerase chain reaction used to direct the replication of any specific DNA sequence chosen by the experimentalist. The fundamental difference between the two methods is that molecular cloning involves replication of the DNA within a living cell, while the polymerase chain reaction replicates DNA in the test tube free of living cells. Formation of recombinant DNA requires a cloning vector, a DNA molecule that replicates within a living cell. Vectors are generally derived from plasmids of viruses and represent relatively small segments of DNA that contain necessary genetic signals for replication, as well as additional elements for convenience in inserting foreign DNA, identifying cells that contain recombinant DNA, and where appropriate, the expression of the foreign DNA. The choice of vectors for molecular cloning depends on the choice of the host organism, the size of the DNA to be cloned, and whether and how the foreign DNA is to be expressed. The DNA segments can be combined by using a variety of methods such as restriction enzyme, ligase cloning, or the Gibson assembly. Expression of recombinant DNA. Following transplantation into the host organism, the foreign DNA contained within the recombinant DNA construct may or may not be expressed. That is, the DNA may simply be replicated without expression, or it may be transcribed and translated so that a recombinant protein is produced. Generally speaking, expression of a foreign gene requires restructuring the gene to include sequences that are required for producing an mRNA molecule that can be used by the host translational apparatus. Example, promoter, translational initiation signal, and transcriptional terminator. Specific changes to the host organism may be made to improve expression of the ectopic gene. In addition, changes may be needed to the coding sequence as well to optimize translation, make the protein soluble, direct the recombinant protein to the proper cellular or intercellular location, and stabilize the protein from degradation. Properties of organisms containing recombinant DNA. In most cases, organisms containing recombinant DNA have apparently normal phenotypes. That is, their appearance, behavior, and metabolism are usually unchanged, and the only way to demonstrate the presence of recombinant sequence is to examine the DNA itself typically using a polymerase chain reaction test. Significant exceptions exist and are discussed below. If the recombinant DNA sequences encode a gene that is expressed, then the presence of RNA and or protein products of the recombinant gene can be detected, typically using the RT-PCR or the Western hybridization methods. Gross phenotypic changes are not the norm unless the recombinant gene has been chosen and modified so as to generate biological activity in the host organism. Additional phenotypes that are encountered include toxicity to the host organism induced by the recombinant gene product, especially if it is overexpressed or expressed within inappropriate cells or tissues. In some cases, recombinant DNA can have deleterious effects even if it is not expressed. One mechanism by which this happens is the insertional inactivation, in which the rDNA becomes inserted into a host cell gene. In some cases, researchers use this phenomenon to knock out genes to determine their biological function and importance. Another mechanism by which the recombinant DNA insertion into chromosomal DNA can affect the gene expression is by inappropriate activation of the previously unexpressed host cell genes. This can happen, for example, when a recombinant DNA fragment containing an active promoter becomes located next to a previously silent host cell gene, or when a host cell gene that functions to restrain gene expression undergoes insertional inactivation by recombinant DNA. Let us now look at applications of recombinant DNA technology. Recombinant DNA is widely used in biotechnology, medicine and research. Today, recombinant proteins and other products that result from the use of recombinant DNA technology can be found in the fields of pharmacy, medical testing laboratory and biological research laboratory. In addition, organisms that have been manipulated using recombinant DNA technology as well as products derived from those organisms have found their way in many farms, supermarkets, home medicines and even pet shops such as those that sell glowfish and other genetically modified animals. The most common application of recombinant DNA is in basic research, in which the technology is important to most current work in the biological and biomedical sciences. Recombinant DNA is used to identify, map and sequence genes and to determine their function. Recombinant DNA probes are employed in analyzing gene expression within individual cells and throughout the tissues of whole organisms. Recombinant proteins are widely used as reagents in laboratory experiments and to generate antibody probes for examining protein synthesis within cells and organisms. Many additional and practical applications of recombinant DNA are found in the industry, food production, human and veterinary medicine, agriculture and bioengineering. Some specific examples could be recombinant chymosin, which is found in rennet, is an enzyme required to manufacture cheese. It was the first genetically engineered food additive that was used commercially. Traditionally, processes obtained chymosin from rennet, a preparation derived from the fourth stomach of the milk-fed calves. Scientists engineered a non-pathogenic strain, K12, of E. coli bacteria for large-scale laboratory production of the enzyme. This microbiologically produced a recombinant enzyme, identical structurally to the calf-derived enzyme, costs less and is produced in abundant quantities. Today, about 60% of the US hard cheese is made with genetically engineered chymosin. In 1990, the FDA granted chymosin generally recognized as safe status based on the data showing that the enzyme was safe. Recombinant human insulin almost completely replaced insulin obtained from animal sources, example from pigs and cattle, for the treatment of insulin-dependent diabetes. A variety of different recombinant insulin preparations are in widespread use. Recombinant insulin is synthesized by inserting the human insulin gene into E. coli, which then produces insulin for human use. Recombinant human growth hormone. This is administered to patients whose pituitary glands secrete insufficient quantities to support normal growth and development. Before recombinant human growth hormone became available, human growth hormone for therapeutic use was obtained from pituitary glands of cadavers. This unsafe practice led to some patients developing Creutzfeldt-Jakob disease. Recombinant human growth hormone eliminated this problem and is now used therapeutically. Recombinant blood clotting factor 8. A blood clotting protein that is administered to patients with forms of the bleeding disorder hemophilia who are unable to produce factor 8 in quantities sufficient to support normal blood coagulation. Before the development of recombinant factor 8, the protein was obtained by processing large quantities of human blood from multiple donors, which carried a very high risk of transmission of blood-borne infectious diseases, for example HIV and hepatitis B. Recombinant hepatitis B vaccine. Hepatitis B infection is controlled through the use of a recombinant hepatitis B vaccine, which contains a form of the hepatitis B virus surface antigen that is produced in yeast cells. The development of the recombinant subunit vaccine was important and necessary development because the hepatitis B virus, unlike other common viruses such as the polio viruses, cannot be grown in vitro. Vaccine information from Hepatitis B Foundation. Diagnosis of infection with HIV. Each of the three widely used methods for diagnosing HIV infection has been developed using recombinant DNA. The antibody test ELISA or Western blot uses a recombinant HIV protein to test for the presence of antibodies that the body has produced in response to an HIV infection. The DNA test looks for the presence of HIV genetic material using a reverse transcriptase polymerase chain reaction. Development of the RT-PCR test was made possible by the molecular cloning and sequence analysis of HIV genomes. Golden rice. A recombinant variety of rice that has been engineered to express the enzymes responsible for the beta-carotene biosynthesis. This variety of rice holds substantial promise for reducing the incidence of vitamin A deficiency in the world's population. Golden rice is not currently in use pending the resolution of regulatory issues. Herbicide resistance crops. Commercial varieties of important agricultural crops including soy, maize, sorghum, canola, alfalfa and cotton have been developed that incorporate a recombinant gene that results in resistance to the herbicide glycophosphate and simplifies weed control by the glycophosphate application. These crops are in common commercial use in several countries. Insect-resistant crops. Bacillus thuringiensis is a bacterium that naturally produces a protein with insecticidal properties. The bacterium has been applied to crop as an insect control strategy for many years, and this practice has been widely adopted in agriculture and gardening. Recently, plants have been developed that express a recombinant form of the bacterial protein, which may effectively control some insect predators. Environmental issues associated with the use of these transgenic crops have not been fully resolved. Let us now look at the different steps in molecular cloning. In standard molecular cloning experiments, the cloning of any DNA fragment essentially involves 7 steps. That is, the first step is choice of a host organism and cloning vector. Step 2, preparation of vector DNA. Step 3, preparation of DNA to be cloned. Step 4, creation of recombinant DNA. Step 5, introduction of recombinant DNA into host organism. Step 6, selection of organisms containing recombinant DNA. Step 7. Screening for clones with desired DNA inserts and biological properties. Choice of host organism and cloning vector. Although a very large number of host organisms and molecular cloning vectors are in use, the great majority of molecular cloning experiments begin with a laboratory strain of the bacterium E. coli, which is the Escherichia coli, and a plasmid cloning vector E. coli, and plasmid vectors in common use because they are technically sophisticated, versatile, widely available, and offer rapid growth of recombinant organisms with minimal equipment. If the DNA to be cloned is exceptionally large, hundreds of thousands to millions of base pairs, then a bacterial artificial chromosome or yeast artificial chromosome vector is often chosen. Specialized applications may call for specialized host vector systems. For example, if the experimentalist wished to harvest a particular protein from a recombinant organism, then an expression vector is chosen that contains appropriate signals for transcription and translation in the desired host organism. Alternatively, if replication of the DNA in different species is desired, for example, transfer of DNA from bacteria to plants, then a multiple host range vector, also termed shuttle vector, may be selected. In practice, however, specialized molecular cloning experiments usually begin with cloning into a bacterial plasmid followed by sub-cloning into a specialized vectors. Whatever combination of host and vector are used, the vector almost always contains 4 DNA segments that are critically important to its function and experimental utility. 1. An origin of DNA replication is necessary for the vector and recombinant sequences linked to it to replicate inside the host organisms. 2. One or more unique restriction endonuclease recognition site that serves as sites where foreign DNA may be introduced. 3. A selectable genetic marker gene that can be used to enable the survival of the cells that have taken up vector sequences. And number 4, an additional gene that can be used for screening with cells containing foreign DNA. Preparation of vector DNA. The cloning vector is treated with a restriction endonuclease to cleave the DNA at the site where foreign DNA will be inserted. The restriction enzyme is chosen to create a configuration at the cleavage site that is compatible with that at the ends of the foreign DNA. Typically, this is done by cleaving the vector DNA and foreign DNA with the same restriction enzyme. Most modern vectors contain a variety of convenient cleavage sites that are unique within the vector molecule, so that the vector can only be cleaved at a single site and is located within a gene whose inactivation can be used to distinguish recombinant from non-recombinant organisms at a later step in the process. To improve the ratio of recombinant to non-recombinant organisms, the cleaved vector may be treated with an enzyme, alkaline phosphatase, that dephosphorylates the vector ends. Vector molecules with dephosphorylated ends are unable to replicate, and replication can only be restored if foreign DNA is integrated into the cleavage site. Preparation of DNA to be cloned. For cloning of genomic DNA, the DNA to be cloned is extracted from the organism of interest. Virtually any tissue source can be used as long as the DNA is not extensively degraded. The DNA is then purified using simple methods to remove contaminating proteins, extraction with phenol, RNA ribonuclease, and smaller molecules. Polymerase chain reaction methods are often used for amplification of specific DNA or RNA sequences prior to molecular cloning. DNA for cloning experiments may also be obtained from RNA using reverse transcriptase, complementary DNA or cDNA cloning, or in the form of synthetic DNA, which could be artificial gene synthesis. cDNA cloning is usually used to obtain clones representative of the mRNA population of the cells of interest. while synthetic DNA is used to obtain any precise sequence defined by the designer. The purified DNA is then treated with a restriction enzyme to generate fragments with ends capable of being linked to those of the vector. If necessary, short double-stranded segments of DNA containing desired restriction sites may be added to create end structures that are compatible with the vector. Creation of recombinant DNA with DNA ligase. The creation of recombinant DNA is in many ways the simplest step of the molecular cloning process. DNA prepared from the vector and foreign source are simply mixed together at an appropriate concentrations and exposed to an enzyme, DNA ligase, that covalently links the ends together. This joining reaction is often termed as ligation. The resulting DNA mixture containing randomly joined ends is then ready for introduction into the host organism. DNA ligase only recognizes and acts on the ends of linear DNA molecules, usually resulting a complex mixture of DNA molecules with randomly joined ends. The desired products, vector DNA covalently linked to foreign DNA, will be present, but other sequences, examples foreign DNA linked to itself, vector DNA linked to itself, and higher order combinations of vector and foreign DNA are also usually present. This complex mixture is sorted out in subsequent steps of the cloning process after the DNA mixture is introduced into cells. Introduction of recombinant DNA into host organism. The DNA mixture previously manipulated in vitro is moved back into a living cell referred to as the host organism. The methods used to get DNA into cells are varied, and the name applied to this step in the molecular cloning process will often depend on the experimental method that is chosen. Example: transformation, transduction, transfection, electroporation. When microorganisms are able to take up and replicate DNA from their local environment, the process is termed transformation. And cells that are in physiological state such that they can take up DNA are said to be the competent. In mammalian cell culture, the analogous process of introducing DNA into cells is commonly termed transfection. Both transformation and transfection usually require preparation of the cells through a special growth regime and chemical treatment process that will vary with the specific species and cell types that are used. Electroporation uses high voltage electrical pulses to translocate DNA across the cell membrane. In contrast, transduction involves the packaging of DNA into virus-derived particles and using these virus-like particles to introduce the encapsulated DNA into the cell through a process resembling viral infection. Although electroporation and transduction are highly specialized methods, they may be the most efficient methods to move DNA into cells. Selection of organisms containing vector sequence. Whichever method is used, the introduction of recombinant DNA into the chosen host organism is usually a low efficiency process. That is, only a small fraction of the cells will actually take up DNA. Experimental scientists deal with this issue through a step of artificial genetic selection, in which cells that have not taken up DNA are selectively killed, and only those cells that can actively replicate DNA containing the selectable marker gene encoded by the vector are able to survive. When bacterial cells are used as host organisms, the selectable marker is usually a gene that confers resistance to an antibiotic that would otherwise kill the cell, typically ampicillin. Cells harboring the plasmid will survive when exposed to the antibiotic, while those that have failed to take up plasmid sequences will die. When mammalian cells are used, a similar strategy is used, except that the marker gene confers resistance to the antibiotic geneticin. screening for clones with desired DNA inserts and biological properties. Modern bacterial cloning vectors use the blue-white screening system to distinguish colonies or clones of transgenic cells from those that contain the parental vector, that is, vector DNA with no recombinant sequence inserted. In these vectors, foreign DNA is inserted into a sequence that encodes an essential part of beta-galactosidase, an enzyme whose activity results in formation of a blue-colored colony on the culture medium that is used for this work. Insertion of the foreign DNA into the beta-galactosidase coding sequence disables the function of the enzymes so that the colonies containing transformed DNA remain colorless, which is white. Therefore, experimentalists are easily able to identify and conduct further studies on transgenic bacterial clones while ignoring those that do not contain recombinant DNA. The total population of individual clones obtained in a molecular cloning experiment is often termed as a DNA library. Libraries may be highly complex, as when cloning complete genomic DNA from an organism, or relatively simple, as when moving a previously cloned DNA fragment into a different plasmid. But it is almost always necessary to examine a number of different clones to be sure that the desired DNA construct is obtained. This may be accomplished through a very wide range of experimental methods, including the use of nucleic acid hybridizations, antibody probes, polymerase chain reactions, restriction fragment analysis, and or DNA sequencing. With this, we come to the end. Thank you.